Welcome to the Science for the Public lecture series. Science for the Public is an organization committed to bringing science information and issues to the general public. Visit our website for our program listings and blog. Welcome to Contemporary Science Issues and Innovations. This evening we have with us two distinguished professors from Tufts University Medical School, Dr. Ana Soto and Dr. Carlos Sonnenschein. They're also professors in the Tufts Sackler School of Graduate Biomedical Sciences. They have been leaders in several areas of medical research, one of which is our topic tonight, endocrine disruptors. Their work in this area is internationally recognized, and they've served on national and international advisory panels on endocrine disruptors. Tonight, Dr. Soto and Sonnenschein discuss the health impact of BPA and other endocrine disruptors widely used in plastics, linings, in food cans, and many other everyday materials. They explain how these chemicals cause such a range of diseases and disorders, and they explain what policies are in place to deal with the problem and what the public must do to ensure protection. In addition, Dr. Soto and Sonnenschein will talk about their very interesting related cancer research, which has led to revolutionary perspectives about cancer. It's a very great honor to welcome these distinguished scientists. Welcome. And may I begin, please, with asking, what does the endocrine system do?
Well, the endocrine system, it's formed by many glands. The glands are little organs, and the cells in those organs produce hormones. Hormones are the mediators of the activity of the endocrine system. So we have the organs that produce the hormones, the endocrine system, and then all around in different parts of the body, there are the cells that respond to these hormones. So the function of the endocrine system is to coordinate different functions in the organism. One of them is to maintain the constancy of the internal milieu, that is the concentration of ions, etc., in the internal fluids. That is the reason why we are not eating constantly. Our nutrients are always present in our serum, etc. The way in which they are utilized and released have to do with hormones. Also, they regulate reproduction and growth of the organism. So there are many functions, and all of them have to be controlled according to the changes in needs as the organism is changing activities.
How do these, which you've called EDCs, what is it that they do that interferes somehow with the endocrine system?
That's right. The reason why endocrine disruptors have taken so important role in our understanding of what's going on in the environment is precisely because they modify all the normal functions that Anna described a minute ago regarding the putting cells and organs in the right conditions to modify, if necessary, our responses. Now, endocrine disruptors precisely prevent some of these events because they compete with normal hormones that are distributed all around, and as a result of this interaction between the hormones or the endocrine disruptors on the target cells that they eventually modify, is that the reason why the different symptoms or disruptions occur. And we have been in the forefront of discovering precisely what are they.
And what are these impacts?
Well,
Like you had many examples, but we see papers constantly now about it's such a diverse impact.
well, precisely. The first ones to be discovered are those that mimic the action of the female hormone estrogens. So by doing that, they interfere with the development of the mammary gland, for example.
Yeah.
They will feminize males. They would alter reproduction, etc. So those are the first kind discovered. And actually the first one probably was DDT. That was discovered, it was given to roosters. And it feminized roosters. So right there, to their surprise, because it's supposedly just an insecticide, it was found to feminize roosters. I think that was 1953. And since then, things have changed. Now we have so many different chemicals, but the problem is that many of them have been used in just packaging, cosmetics, and practically everything we are in contact with.
And that includes
Will
like
be
cans, everything.
Yes, like soup cans or etc. And so we are exposed to these chemicals through our food, our water, etc. And our air too. And like indoor air could contain many of these chemicals.
Our contribution precisely has been that early on we bumped into an accident in the laboratory that allowed us to accidentally discover that certain chemicals, specifically nonylphenol, interfere with the effect of estrogens. And it was in 1989 that we faced this interesting problem in the laboratory, whereby the experiment that we were running eventually ran amok, and we didn't know why until after a long process of inquiry, we discovered that this was precisely due to the fact that certain chemicals, specifically nonylphenol, were shed from the centrifuge tubes that we were using in our research.
And so it's kind of an accident, but you, as it turned out, it really gave you a big lead. Was this the test tube, the famous test tube accident?
We were in a way prepared because we were studying the effects of the ovarian hormone, estradiol, and in our assay, you would expect that only cells that have been exposed to estradiol, the female hormone, will proliferate. So we were expecting huge differences in cell numbers between the controls that didn't have the hormone and the ones that had the hormone.
Right.
All of a sudden, everything, every culture had too many cells. And we knew that the only chemical we knew that could do that was estrogen.
So you were all prepared, but it was still a surprise to find this in
4 months,
test tubes.
4 months to figure out where it was coming from.
Right.
And then 1 year to purify it from plastic. I mean, we had to extract innumerable number of tubes. I mean, like hundreds of them in order to extract sufficient material to determine the chemical nature of it.
Did I read that you actually had to inform the test tube manufacturer that you learned about it before they learned about it?
Right.
Right.
This is precisely one of the characteristics of these endocrine disruptors. People are not prevented from manufacturing all kinds of products without exactly knowing what is involved in it. And when we called the manufacturer of these tubes, first of all, they couldn't, let's say, believe it, and then they accepted they didn't want to tell us what it was because of proprietary reasons. So they sent us several batches of these centrifuge tubes that were manufactured before and after we had the accident. And they said, yes, we now understand why this has happened, because the overt intention was to improve on the product.
Right.
Because these centrifuge tubes were very friable. They could break very easily.
Right.
So they improved it.
Yeah.
And in the process of improving, they added this. They didn't change the catalog number. They didn't change anything, assuming that this was just a issue that did not pertain to our knowledge, the knowledge of the user.
Exactly, exactly. So it was a kind of a multiple breakthrough there,
right.
But it must have been important for other labs as well, because nobody knew this. You were doing a kind of unique research, I suppose, that got it started, but that's quite a remarkable story.
It's interesting because some other groups rediscover it later on, and so it affected other people as well. But if you are not working with estrogens, you won't notice.
So you were able to beat everybody to it. But I wanted to ask you about this very broad impact on health, just so many things, obesity and cancer, which we're coming back to, and even things, cognitive things, possibly autism and stuff. Are you all convinced, before we go on, that it is that widespread? That
if you see the animal studies,
okay.
you cannot doubt that those effects take place. The animals show, and now we are talking about exposures during development, fetal development, and clearly the animals become obese. One of the most interesting things, now I'm talking exclusively about bisphenol A, that is also a plastic, a plastic estrogen, and it's clear that animals exposed developmentally later on develop obesity, alterations in behavior like anxiety, that people have linked them with malattention syndromes, if you will. Behaviors, repetitive behaviors that also researchers think are the mice and rat equivalent of autism.
Yes.
Decreased reproduction.
Yeah.
The flippers. Decreased reproduction, alterations in reproduction. Early cessation of cyclical activity of the ovary. It would be like getting early menopause.
Yeah.
If that happens in women, that would mean an increase of the risk of heart disease early on. The latest discovery was that it affects, BPA also affects the heart in females. And then, of course, the work on cancer, that it increases the propensity to prostate cancer, to develop prostate cancer and breast cancer.
Yeah, it's interesting you mentioned whether we were sure of this, and precisely it's difficult to be sure. We do experimental biology precisely to see whether there is an issue with animals that can be extrapolated to humans. Sometimes people say, but you haven't proved it in humans. Of course, we would need volunteers to be exposed to this.
And
that is obviously an ethical problem that have to be recognized. So what we do is to extrapolate in the closest possible way with what happened in animals to what may happen in humans. And eventually it happens in humans, as have been shown in the European Union with some research done on that subject.
Right.
Yeah.
I want to come back to this and on the cancer stuff that you've really been the leaders in. But I want to ask you before we go on, I want to get back to the generational thing. So you've done a lot of work on the effects on the embryo, and you have shown that it affects, it's not limited to the generation at hand. Can you explain that for us?
Okay, there are two things. One is that the effects, I'm saying that because I'm showing a slide.
Okay.
And the slide shows you our history. We start being one cell, becomes 2, 4, etc. As you see in the screen, there are no organs, nothing to discern at the beginning. So the whole process after up to birth is becoming all this creation of organs.
Right.
And so if you expose animals or humans during fetal development, the effect will be irreversible, because you will be affecting the formation of an organ and the function, therefore. Whereas if you expose humans or animals at adult age, where, you know, once those organs are formed, it's not that it would produce no effects, but a good deal of those effects will be reversible. So that is the huge difference between being exposed as a fetus and being exposed as an adult. Now, the other question is about very early exposure and how that affects the germ cells. Would you like to explain transgeneration?
As the slide shows, we are talking about the fragile fetus precisely because it is at that time when the organs are being formed, and as a result of these interactions between the endocrine disruptors and the formation of these organs, some malformations occur, and for the most part they take effect in organs that are target for these estrogen, especially the estrogenic endocrine disruptors. So as a result, what we see when these animals, these fetuses are exposed and wait until they become adults, we see the effect that have been, let's say, kept silence until the function shows up, as for example, in the case of DES, the diethylstilbestrol, the cancer of the vagina appears when these women that were exposed during pregnancy, they are shown in puberty as having these cancers.
Which would be very extraordinary to see a lot of cancer at that age, right?
No, yeah,
so, but it's just as that matures.
Yes, it was 6 cases reported at Mass General Hospital in 1971. So people were exposed, pregnant women were exposed between 48 and 71. It was stopped because the girls, just very young women, got clear cell carcinomas of the vagina.
And that must have been really extraordinary. That seems so young. You don't think of that kind of cancer, reproductive organ cancers, that young. Very amazing, yeah.
That would appear in non-exposed people at 60 and more.
Yes,
and it's not a very frequent cancer. But if we go back to the transgenerational, if you expose animals very early during the period of formation of the gonads, so you see, before we become, of course we have a chromosomal sex, but then early on you cannot distinguish a female from a male. There is a process whereby the gonad will become male or female. When animals or humans are exposed during that period, then you would affect a process that is going on the gametes, that is the cells that will form the next generation.
I see.
And that process is not, it has nothing to do with genes, in the sense that it doesn't alter the genes, but it's an epigenetic process. In other words, those, the genes are marked indirectly at that time. That is normal. It's marked physiologically, but if an endocrine disruptor is present, the marks will happen somewhere else. And that has the propensity to be very stable. I mean, that type of marking. So in animal studies, that the animals were followed up to the 4th generation, the malformations, the defects, the deficits could be observed in the 1st, 2nd, 3rd, and 4th generation. So it's quite stable, and that is why it's a cause of concern.
Absolutely, because it could doom a whole species if it's affecting reproduction organs alone. Then, as we saw with like amphibians very early on, that's very worrisome. But another thing here is we're talking about trace amounts, aren't we? That was one of your points, I believe, that we're not talking about large amounts of this stuff, even very small amounts. Could you elaborate a little on that?
The idea has been always, from a toxicological point of view, that almost anything that is administered in high concentrations will produce toxic effects. Now, with this advent of endocrine disruptors, we now know that there is what is called an inverted U curve, whereby small and high concentrations produce comparable effects. Very low concentrations are indeed producing many of these effects precisely because the sensitive nature of the cells that are affected by it.
Now, the other sort of a corollary there is that even if you could rid the environment, stop producing these things, somehow get them out of everything they're in, which is practically everything now, then even then, well, first of all, can you get rid of them that easily? The second thing is you're still dealing with a generational impact, and nobody knows maybe where that's going to lead. Is that?
Exactly. So you have the good news and the bad news.
Okay.
The good news is, it's again an experiment carried out here in Massachusetts, Silent Spring Institute. So what they did is they provided volunteers with food. So they had to eat canned food for a while, and so they measure bisphenol A in their urine. And then they give them clean, healthy, homemade food for a while. I think it was a few days. And you could see how significant the decrease of bisphenol A in urine was. Now, this is a case in which you can act because bisphenol A is more or less rapidly metabolized. If you find a way to remove it from the diet. Normally we are exposed every day, so we always have a level. So with some chemicals you can control it quite rapidly. Now, if a transgenerational effect already happened, there is not much you can do, but at least you can stop exposing people and producing some of those effects. For chemicals that have a very long half-life, it's a different story. Like, for example, PCBs have a half-life of 30 years. That is that your level in blood will decrease to half of what you have at birth by the time you are 30. In that case, the effect won't be seen that soon. In any case, I think, in both cases, I think it's worth a try.
Oh, yes, absolutely.
Incidentally, the studies that were made by the CDC, this is the Center for Disease Control, indicate that 82% of the population of the United States have high levels of endocrine disruptors, specifically the BPA. This is, I said 82, but 92%. So this gives you a magnitude of the problem.
It really is a huge problem,
And as a result, it is difficult to find controls when people wish to establish epidemiologically what is happening. In other words, you say, well, we'll take this population that has been exposed versus this population that has not been exposed. In fact, all of us are exposed.
Exactly. And it's ubiquitous. It's all around us. It's very hard. So you have an experiment where you give people healthy food, at least for a while, but it's still out there. So it's very hard. I'd like to delve into that a little more, because you come up with clever ways at least to get into it. But while you're on that point, if it is rather urgent to take care of this. You are experienced with countries all over the world and their respective policies, and the policy in the United States. It's my impression that the FDA has been really dragging its feet. So I would like to ask you about the policies for what are people doing around the world. And
the issue, you are right, there are different ways to deal with the problem, and for the most part, the FDA is reluctant to have, not only the FDA, many state offices are reluctant to deal as should be done in preventing the exposure of especially children and mothers that are pregnant with these compounds. This requires a political commitment on the part of the regulators, and usually regulators are not very sensitive to those requests for a number of reasons. There are countries where our plea to act has been listened more, and there have been even states here in the United States that have been more sensitive than others. But it requires this type of political action that would put people, make people aware that this is a serious problem.
Are there countries where that you consider have a good policy about this? They're trying to do something in terms of regulating. Is there a big difference?
Well, there is a difference. Like in the European Union, there is the REACH, and they are going to start testing chemicals, among other things, for endocrine disruption. It's not ideal. Nothing is ideal because, as you may imagine, there are many interests in play. But it's better than here. Starting with the idea that they accept the precautionary principle, and using that, they had removed phthalates, and which has been a very good idea. What is very remarkable is that most of the action occur because of public outcry, and it was through the legislators, not through the organisms that are supposed to regulate. That is the case of the equivalent of FDA in Europe, that is called EFSA.
I see.
It's doing nothing, but there is this regulation that comes from legislation. I mean, the directive came because of legislative action in France, in Denmark, and in Germany. Okay.
But you have a public there in those countries that is well known for being well informed and assertive about policies they want to put through. Does that make a difference somehow?
Yes.
Probably.
And also legislators that will hear
Yes.
people like us that would be interested in talking to people that that know science. I mean, there is a more science-friendly environment
In this.
In addition, because of, let's say, the characteristic of the United States and the European Union, there are a lot of legislators who are MDs, medical doctors, and they are more sensitive to what eventually may happen to their constituents.
Right.
So that is another factor that has influenced a more proactive, together with the precautionary principle that has passed, which here is far from...
Right. And you have the additional problem perhaps in the United States where each state can regulate things off and on. So you have whole blocks of states that will be antagonistic. But the problem with something like this is that the stuff is throughout the environment. It's affecting everything, and people are moving around and so on. So the fact that they won't regulate in state X means that it's going to affect a state that has a better conscience about it, for instance. So it's a big problem, evidently.
Unfortunately, for example, now there are states that are more aware that there is such a thing called global warming.
Yes.
Because of personal events that happen nearby.
Right.
So I hope that the states will now be more aware of the fact that this is not a free thing, that using all kinds of hydrocarbons to produce heat and things like that. We have to renew our views in that regard.
Right. If we could put a halt, if we could control things abruptly now, have we reached like a tipping point with, has come up with climate? You know, that you reach a point where things are already accelerating so fast that you have to come up with other kinds of solutions, not just stopping whatever this problem is.
We think that there is a very serious problem here, and I would say that we are already taking societies, trying to fix a problem that already exists. For example, in Denmark, it's clear that there is a huge increase on reproductive, assisted reproduction. So when they did the study, there they have single payer, and that is the other story. In Europe, they have single payer.
Right.
You can do
universal health care.
Exactly. So you can go to the data set and see what's going on. And so therefore you can see those changes quite rapidly. And they have found that it's about 6% of people that require assisted reproduction, a large increase. And people would say, well, but it wasn't available 30 years ago.
Yeah.
But they did something quite interesting, that is to look by age. Okay, so who's looking for assisted reproduction? The youngest people.
Very
interesting. Not the oldest people.
Right.
So there is a difference, and that was first observed with a decrease on the quality of sperm in Denmark.
Right. Yes.
That was published in the early 90s. So we have a serious problem there, because if we do not reproduce, well, the species will be extinct. And the problem seems to be there. Now, whether we are optimistic or not, I think that we have to act anyway. I don't think that is totally reversible and it's totally, I don't think that we reach that point, but I don't know if we keep doing nothing, whether that will happen in 10 years. I have no idea. I know that it's a serious problem that requires action.
Right.
In the same vein of my previous comment, originally it was thought that this was a Danish problem.
The fertility problem.
Yes.
Only the Danes.
Yes. So then the French verified that that was a problem there. Now in Israel, they have published just this last week or two a paper in which there is a problem with the viability of sperm in Israel. So people will start reacting when it touches them directly.
Exactly.
So in that sense, we are hopeful. I hope that it will not be too late. But this is the reality. People try not to see or not to hear, because obviously these are bad news.
Right.
Nobody wants to listen to bad news.
Right. I am very eager to look at your research. This is, I don't know if people are aware that you had a number of big breakthroughs, and I don't think that in the public we're very aware of what goes into research, how long it takes, the difficulties, especially of doing assays, and maybe you'll explain that a little bit.
Regarding our own experience, which is the experience of probably a lot of researchers, is that you enter a field under the assumption that what you are being told or what you read is, let's say, reality. So you enter the field with trying to expose other issues. Eventually, this shows, as in our case, that what we thought was accepted is not. Specifically, as we started working in control of cell proliferation using estrogens as the, let's say, the lever that would indicate how cells proliferate, we realized that what the textbooks say and what the publications in general say may not be the reality. It is true that you bump into problems when you start a new type of research. As we published, for example, the first cell line that was targeted for estrogens. And as a result, we asked the questions using cells in culture. Now, perhaps Anna could follow that
Well, yeah,
trend.
I would say when this started, the idea was that we know that if you take an animal, female, and you remove the ovaries, the uterus atrophies, becomes smaller. So you give estrogens and then it will become bigger, it contains more cells. Now, when you give estrogens, what you are doing is try to recapitulate what nature is doing in every menstrual cycle, that is making the uterus prepare for implantation. Okay, so one part of it is that more cells are being produced, and estrogens in the animal seem to do that, to make cells produce more cells. So we were under the impression that estrogens would stimulate the production of cells directly. And what we observed once the cell line was established was that those cells will proliferate maximally in the presence and in the absence of estrogens. In culture. So that was totally different than what you saw in the animal. Now, why was that? We put the cells back, because this is a culture, so you can inject them back into the animal, and in the animal, they needed estrogens. Okay, so there was something, a link, that was present in the animal but wasn't present in the culture, and we decided that that could be, we speculated that that was something like a hormone, something that would be contained in serum. So what we did is we now put the cells in contact with medium containing serum, and they didn't proliferate. And only proliferated when you put estrogens. So that was quite interesting because, and it could have ended there, because then we say, well, okay, estradiol is not stimulating, but is blocking the action of an inhibitor, which is totally different. But then you think of it and you realize that all these microbiologists that are working with animals that are unicellular have said always that you don't have to stimulate cell proliferation. If you put bacteria anywhere, well, as far as there are nutrients, they will proliferate.
Exactly.
So that is defined as the default state. That is, what do they do when they have nutrients? And when they do have nutrients, they proliferate. A famous Nobel laureate, François Jacob, used to say the dream of a bacteria is to become two. Okay, so they do proliferate, and nobody or nothing stimulates them. So now we reunite these two things, because what it showed to us is that the default state continues being the same in metazoa, but multicellular organisms. The difference being that the organism is regulating that by putting inhibitors.
In other words, we have a shape due to the fact precisely to this regulation by inhibitors. Now, there is another important argument, which is an evolutionary argument, that supposedly this the original cell that is responsible for all of us being here,
Luca,
that cell didn't have around anyone who would tell it or stimulate it to proliferate. So that is a so-called epistemological argument based on evolution that indicates that there is no need for cells to be stimulated. Proliferation will occur whenever you have these nutrients. And that original cell, in addition to proliferate as a default, moved as a default. In other words, didn't have to be stimulated to move. It already had that intrinsic property within it. And as a result, due to evolutionary mechanisms, it is maintained in multicellular organisms like us. So when people say that in order to a cell to metastasize has to be stimulated to go places, cells move and proliferate just because there is that ability that they carry with them.
I see. Right.
So the only thing you can do to regulate proliferation is to stop it. That is one of the things that was, let's say, a way to tell us that you have to be careful with what textbooks say.
And so this came out of your experience. Fortunately, you were sort of prepared mentally for the discovery, but nevertheless, there was a kind of inherent contradiction to everything that you had
It was a paradox.
schooled. Exactly.
And as Niels Bohr said,
Yes.
you always are looking to bump into a paradox,
Yes,
because
that's right.
behind a paradox there is an important thing.
That's exactly right, and you find it all the time, so you're ready for that in there. Now, in your work, you set up a very important, and I guess it's the assay, is that correct, that everybody now uses, and you're cited all the time for this, and we don't know what e-screen is, but are you able to explain it to us a little bit that we can understand?
So the e-screen, remember we talked about the first estrogen from plastic, nonylphenol, that was discovered in our laboratory. So we adopted this phenomenon, the phenomenon of being hit by these unknown estrogens, and develop it into a screen to detect estrogens. So what we did was, now we have a negative control, cells, estrogen-sensitive cells, in the presence of serum do not proliferate.
Okay, right.
Negative control.
Right.
Positive control, in the presence of serum plus estrogens, they proliferate about 10-fold more in our conditions, okay? So you see 10-fold more cells. Fine. So now you take any chemical that you wish to test and you add to medium that doesn't contain estrogen, and you look and see whether you get few cells or lots of cells. If you got lots of cells, this is an estrogen. And so using this method is very simple. It can be done in culture, and most of the chemicals that were found to be estrogenic after nonylphenol were found to be estrogenic using the e-screen assay. Actually, what happened is that right after we discovered nonylphenol, Theo Colborn organized the Windspread Conference that took place in 1991. And she invited me because one of the elements that was missing there, I mean, she was interested in the phenotype of animals in the Great Lake. The animals were feminized, remember, and they have malformations. So, but if that was due to chemicals that were already banned, we just had to sit and wait. So she found out that Carlos and I had discovered nonylphenol, and that wasn't regulated. So that was like a molecule that demonstrated that the problem wasn't a solved problem, that we had to look into this. And when I was at Winspread, Pete Myers, that was then the president of W.L. Alton Jones, thought that that was a great assay, and gave us a grant, a very small grant, but nonetheless with it, we were able to identify about 15, 20 compounds that were estrogenic among common things that are used in the environment, pesticides, chemicals that are in plastic, etc.
Now, you have to keep in mind that at this point, I think that there are 150 compounds that have been characterized, but there are 80,000 that are in current use that have not been tested.
Mm-hmm.
Oh dear, right.
Oh dear.
Okay.
Right. And then we're talking about things that pervade air, water, everything, all of your food and everything else. So you have a lot of work to do, don't you? Before, as we're moving into close down time here, I'm afraid, but I wonder if you could tell us a bit about the cancer, this just turned things around. I'm very eager to find out.
Again,
what the status of it is now.
Again, this episode that we are talking about occurred in the late 80s, early 90s. At that time, we started thinking that it would be a good idea to put together whatever we have learned into a book, always thinking that, as the textbooks even today claim, that cancer was a problem of cell proliferation. And in 1993, we started writing the book under the impression that indeed cell proliferation was at the center of cancer. And we thought, now that we understand how cell proliferation occurs, we will understand what cancer is or how it evolves. And we, now in retrospect, can claim that we were wrong. The problem of cancer is not a problem of control of cell proliferation. The problem of cancer is a much more complex issue that involves the interaction of tissues and not of individual cells. In other words, the center is not a cell that goes haywire.
Which is what we're taught.
Right.
And all the
And
textbooks will say this, and whole
and
institutions are working on it.
It's a genetic-based event, a cell-based event. So we started thinking, and again, we bump into evidence that indicated precisely that the problem is a problem of tissues that interact in the wrong way.
I see.
And actually, I think that in a way we can say that the two things merge there.
Yes,
the endocrine disruptors with our work in cancer.
Right.
Because there we realized that cancer was a problem of organogenesis, of histogenesis, of the formation of structures, shapes, organs, where many cell types collaborate. So we say that cancer is development gone array. So what we did is we work with the mammary gland. So what we did is to separate what is called the support tissue or stroma.
Yes.
And the epithelium, that is the business part of the gland. Okay, so the business part that produces milk. Okay, so we separated the two tissues and exposed them separately to a carcinogen, or the buffer in which the carcinogen was dissolved. So now you have four combinations. Stroma exposed, epithelium not exposed, epithelium exposed, stroma not exposed, both of them exposed, both of them non-exposed. Right. So you look at the four combinations, and what do you find? We found that if you expose the support tissue, the stroma, and not the epithelium, cancer arose. And that is totally contrary
to
the somatic mutation theory, that is the theory of record, because in their case, the cell that gives name to the tumor, that is the epithelial cell, should be the one that reacts to the carcinogen, becoming a cancer cell. Our experiment showed that, to the contrary, what is going on there is that you need the interaction of many cell types to get to this formation of a tumor. And we ran the converse experiment,
right,
which is again the same thing. Now, you take a cancer and you separate the epithelial cells, and then you inject them in normal stroma. And what you find is that a normal stroma will inhibit those cells, but not only to inhibit them from proliferating, it will push those cells to form normal structures. So you get normalization of what once was tissue, cancer tissue. And we are not the only ones that have shown that. I mean, right now there is about probably 30 references where people have done that with all kinds of tissues. So cancer can be reversed by normal environments.
Right, which is something that the somatic mutation theory does not consider.
Yes.
In other words, mutations are stable, and as a result, whatever happened, happened. And this is going to be the end of it, the story. While if you consider that cancer is a reversible issue, you have to explain how come that this reversible process occurs. So we have been able to produce a tumor without exposing the cell that is going to characterize the tumor as a carcinoma. We exposed the stroma only, and we got a carcinoma. And when we got a carcinoma, we could normalize it by putting it in contact with a normal stroma.
Now, this is a sort of giant leap forward. Did you find resistance to this? It's so typical in the history of science, isn't it, that somebody finds the right answer to something and they're like, no, it's not right.
But that is, as you said, this is the norm. In other words, first of all, they say that it cannot happen. Then they say, well, maybe. And finally, they say, we knew it all along.
Yeah, of course, there you are.
we are at this point almost to the point in which there are people who are starting to believe that there is something that we have said. By the way, our book was published in 1998. So it has already, let's say, over a decade of being exposed. And of course, we kept on doing research and publishing on the subject. And we are invited to talk to many fora here in the States as well as in all over the world. So we try to convince people that this represents a real change, not only from the point of view of the theory itself that we call the tissue organization field theory, but the prognostic of cancer.
I should say.
In other words, we are being told every year that in the next 10 years there would be a solution to the problem. And year after year, these 10 years are going forward, extended in such a way that people are starting to believe that there is something wrong with the way we look at cancer. We are proposing precisely to look at cancer in a different way that we and others consider that is more realistic.
Right. Well, I'm not an expert, but I consider the evidence you brought was very clear.
At the same time, there was Max Planck who said that
That's true.
You don't convince people with data. You expect that eventually they will die.
So new philosophers are listened to when the old philosophers die.
It's exactly true.
So you see, there is a sociology of science.
There really is, and that's your next book, perhaps, that
Yes,
I
we
would
are.
like to, because
Let's see.
I had the opportunity to speak with the two of you very briefly about this, about the philosophy of science, which
Yes.
we're not given at all, so we don't have a sense usually of the history of the thing. What is your advice for citizens in this country that way?
Well, I think that citizens have to understand the basics of science, because otherwise they are going to be told things that are not true, and they would have no way to decide. I think that Ernst Mayr, in the prologue to his book, This is Biology, he said that he wrote that book precisely to educate people, because you only can have a democracy if you have citizens that are well informed. So we need to increase education in science, and I think that Carlos will expand on that, but I think that it's not only science. I think that we have to go a little bit back in time and think the way people were educated in the 50s, where people had to know history, philosophy, and all that, what is called a liberal math, liberal education, because you need to know history, you need to know
in order to see the picture.
You have to see the big picture, and I think that education has to start very early in that regard. But Carlos has more ideas.
Well, of course, science was invented by human beings.
Yes.
And it has a tradition of about 500 years, since the Renaissance. And science is a social event. And people should recognize as well, in order to make them, let's say, come to scientists, that we are part of the society to which they belong. And unless you encourage them and you tell them things as they are, things that we don't know, we will get more credibility, because unfortunately, for whatever reason, we are creating a group of people who have become the high priests of even religion, when in fact we don't have the right to do that. We believe that scientists are part of this society, and they should be asked the same questions that we ask bankers, unfortunately with certain
Try to get them to
results,
talk to the people, right?
and politicians,
Right.
and all the people who claim that have the right to clarify our ideas.
Right.
And scientists are not an exception.
Right. So we need a dialogue, but
Absolutely.
an informed dialogue. I do appreciate very much your coming in and talking with us, and it's been most interesting for me. Thank you so much.
Thank you for
Thank you
giving us
for
the
having
opportunity.
us.